ATP, cellular energy currency. ATP is what the body uses for energy. Okay. Adenosine triphosphate. That's what we exchange energy for. When it breaks down, it goes from ATP to ADP, adenosine diphosphate. There's two phosphate groups, and energy is given off. So catabolism is the metabolic pathway that break down complex macromolecules into smaller ones. It's a process of harvest parts of potential energy for use by the cell. So catabolism is when we ingest something, we break it down, and then we can use that to produce energy that can be stored in the body for things that we need to do that require energy. Okay. One energy requiring function is anabolism. Anabolism is making it, putting things together, making complex molecules. Okay. It's biosynthesis. It's the ability to make complex molecules. Catabolism breaks them down. Anabolism builds it back up. Right? Cells use an energy conversion strategy that oxidizes glucose. Okay, so we oxidize glucose to produce ATP. And we'll show you how we run that through glycolysis and through the Krebs cycle and how we use that to produce ATP in the body. So small amounts of energy are released at several points in this pathway as glucose is broken down. The energy is harvested and stored in the bonds of ATP. So it's stored in the bonds of adenosine triphosphate. It has 3 phosphate groups, and those are high energy bonds. ATP is a universal energy currency. That's what we use to store energy. That's what gives us our energy. Adenosine triphosphate. Okay, types of cell work that require energy. Okay, like I said, building things, biosynthesis. Okay, biosynthesis, glucose from CO2 and water. That occurs in photosynthesis in plants. Plants take in CO2 and water, they produce and they form glucose and oxygen as a byproduct. It takes energy to do that. Okay. But plants use the sun's energy for that to accomplish. Synthesis of amino acids that the body can make. Synthesis of nucleotides for DNA and RNA. Synthesis of lipids, of our fat storage. Protein synthesis from amino acids, as we use our amino acids to link them with transfer RNA, which you just talked about, to form proteins. Synthesis of nucleic acid, making of DNA and RNA. Synthesis of organelles and membranes around the cell to keep the cell functioning. We talked about their active transport. Active transport is the movement of things across the cell membrane. You have to get things into the cell, get things out of the cell for a cell to function. Transport H+ to maintain a constant pH. The body has to maintain a certain temperature and a certain pH for all these enzymes to work. Okay, the pH has to stay between 7.3 to 7.4, or enzymes are going to cease to function. We have to transport food molecules into cells, nutrients. Transport sodium, potassium. That's how nerves work. Okay? We have to exchange sodium, potassium across that membrane for that nerve impulse to travel. Okay? That's if you ever go to the dentist and we give an injection to get you numb, right? That's not stopping the pain. All we're injecting into is a salt blocker. It blocks salt. It stops sodium and potassium trading places. So the pain's still there. It's just the nerve that fires it to tell the brain it's painful. We've stopped the salt. We've stopped the movement of sodium potassium ions. We've stopped salt. So the brain doesn't send a message to get that's painful. That's all local anesthetic is. Okay. Secretion of hydrochloric acid into the stomach for breaking down foods. Transportation of waste into the bloodstream, into the urine, into the kidneys. Transport amino acids to the sugars in the bloodstream from the intestines, and accumulation of calcium ions in the mitochondria. We have to have calcium ions. There's 2 things that every muscle cell requires to contract. That's calcium and ATP. You have to have both those. Without both of those, your muscle cells are not going to contract. And your heart's a muscle. So without calcium and ATP, your heart stops. You have to have those for all muscles in the body, calcium and ATP. Motility, okay? Contraction and flexing muscle cells. Like I said, contracting muscles, flexing muscles, all require ATP, require energy. Separating chromosomes during cell division, ability of sperm to swim, like through their flagella. Movement of foreign substance out of the respiratory tract by the cilia that beat in one direction so everything comes up when you breathe pathogens into your lungs. translocation of eggs in the fallopian tubes, so females can get pregnant in the reproductive tract. Okay, all these things require energy. They have to have energy in the form of ATP. Okay, ATP is adenosine triphosphate, 3 phosphate groups. Okay, it serves as a go-between molecule that couples things together, couples exergonic and endergonic reactions. Exergonic releases energy. Okay, when you break things down, energy is released. Endergonic reactions, okay, energy requiring. This is anabolic reaction. That's building things up. When you have to build something up, it requires energy. Breaking it down gives off energy. So ATP captures energy as a phosphoanhydride bond. Okay? The hydrolysis of the anhydride bond provides energy for anabolism. So once we capture it, we can use that energy to build what we need in the body. AP is nucleotide, adenosine. It's a molecule of a nitrogenous base, which is adenosine, a 5-carbon sugar, and has 1, 2, or 3 phosphoryl groups. So adenosine monophosphate is 1 group. Adenosine diphosphate is 2 phosphoryl groups. Adenosine triphosphate is 3 phosphoryl groups. So the phosphoester bond joins the first phosphoryl group to the 5-carbon sugar ribose. Okay, just like it did in DNA, building the sugar phosphate backbone. Probably on the word there. Okay. The second and third groups are bound by a phosphoanhydride bond, which are high energy bonds. Okay. So your phosphoanhydride bond joins the last ATP, the last phosphate group, and the second last one. Okay. So there's what it looks like. There's adenosine. Here's your nitrogenous base. Here's your sugar, your ribose sugar. And this is 1, 2, 3. The last 2 phosphate groups are joined by the phosphoanhydride bond. Those are very high energy bonds, release a lot of energy when they are broken. So you have adenosine, adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate. Okay. So hydrolysis of the phosphoanhydride bond, when it gets broken, when these phosphoanhydride bonds are broken, large amounts of energy are released. Okay. This energy can be used for cellular work. It can drive the cellular process, like phosphorylation of glucose. We have to phosphorylate glucose before it can enter into the Krebs cycle. And we break it down to produce energy. All right. So we start with beta-D-glucose plus adenosine triphosphate, okay, plus water. Okay. You can hydrolyze that reaction. Okay. You have adenosine triphosphate. You have beta-D-glucose 6-phosphate. So we took one of the phosphate groups and put it onto the glucose molecule. Okay. So we have adenosine diphosphate, ADP. We have ATP. And we took this phosphate group and we transferred that group onto the glucose molecule. We phosphorylated the glucose molecule. Now we can use the glucose molecule in the Krebs cycle and start the breakdown process of it. Okay. And all we did was add water between the 2 groups. Okay. So we took water and deposited that between it. We have H and OH, and allows that to hydrolyze that bond. Okay. So step 1 is hydrolysis. ATP plus water goes to ADP plus your phosphate group, and it releases 7 kilocalories per mole of energy. Then we want to take that energy that we've used and we want to form glucose 6-phosphate, G6P, glucose 6-phosphate. So we take 30 kilocalories of that energy, react with glucose, which is C6H12O6, plus our phosphate group to give us glucose 6-phosphate, and then water is given off between that when the bond is formed. We lose an H from the phosphate group, OH from the glucose, and we form that linkage, that glucose 6-phosphate. So if you take those 2 combinations, you take those 2 equations and add them together, Okay, you look at if you look like there's water on this side, water on this side, that's about it. Okay, so we take glucose plus ATP, which here's our glucose, and our ATP goes to glucose 6-phosphate plus ADP, and we have 7 kilocalories when the bond was broken, and 3 kilocalories was used to make it. So we have an abundance or leftover of 4 kilocalorie energy being released. So it's an overall energy releasing process. So it proceeds spontaneously because it gives off energy. It doesn't take energy, it produces energy. So it can occur spontaneously. We don't have to put energy in to make it happen. So it's 3 steps. You got hydrolysis of glucose, hydrolysis of ATP, breaks the phosphate bond. You synthesize glucose 6-phosphate from glucose. You add the phosphate group to it. Okay. And you add those 2 reactions over, add those 2 together. Yeah, glucose plus ATP goes to glucose 6-phosphate plus ADP now, and 4 kilocalories per mole of energy. Okay, so why is ATP called the universal energy currency? Because we store it. We use it for energy storage. Okay, it's the major molecule used by all organisms to store energy, not just humans. Okay, lists 5 biological activities that require ATP. Anytime you're building something, you want to have biosynthesis, it is going to require energy. Okay, muscle contraction, which we talked about, protein synthesis, lipid synthesis, DNA synthesis, and nerve conduction. Those are some of the things we talked about. Okay, there's a whole lot of others. Okay. So the catabolic process is a process of breaking down. Carbohydrates, fats, and proteins can be degraded to release energy. Remember, catabolic processes release energy. You break them apart, you're breaking chemical bonds, energy is released. You're building them up, you're forming chemical bonds, energy is absorbed. Okay. Carbohydrates are the most readily used energy source that we have. Okay. So we take in food at the top of the chain here. Okay. Food consists of proteins, carbohydrates, and fats. Okay. We break down proteins to amino acids. We break down carbohydrates to simple sugars. We break down fats to fatty acids and glycerol. We talked about glycerol, the 3-carbon alcohol that allows you to put long-chain fatty acids attached to it. All right, so the amino acids break down, and they can go into pyruvate, they can go to acetyl coenzyme A, and they can enter the citric acid cycle. And when this cycle goes through, it produces a large amount of ATP, the citric acid cycle here. The simple sugars undergo glycolysis. You add phosphate to glucose. So you start breaking down glucose, and you release ATP. Glucose breaks down into pyruvate, which is a 3-carbon molecule. Breaks that down to acetyl coenzyme A, enters the citric acid cycle. Fatty acids go to acetyl coenzyme A, enter the citric acid cycle. So every one of those food groups, whether it's going to be proteins, carbohydrates, or fats, is going to enter the citric acid cycle. Oxidative phosphorylation is oxidation of acetyl coenzyme A, and that produces ATP. And we show how much it's going to produce depending on how much what we put into it. Okay. So stage 1 is hydrolysis of macromolecules to subunits. We break them apart. Proteins break to amino acids. Carbohydrates break down to simple sugars. Fats break down to fatty acids and glycerol. Okay. Then step 2, conversion of these subunits to form that can be completely oxidized. Usually it goes to either pyruvate and then to acetyl coenzyme A. We can completely oxidize acetyl coenzyme A to carbon dioxide in the citric acid cycle and produce a lot of ATP in the process. Stage 3 is the complete oxidation of acetyl coenzyme A and the production of ATP. That's your 3 steps. Okay. So the first stage, hydrolysis of dietary macromolecules to smaller subunits. So the purpose of stage 1 in catabolism, remember catabolism is the breaking down of either proteins, lipids, or carbohydrates. So the purpose of stage 1 in catabolism is to degrade food into their component subunits. Okay. Polysaccharides are degraded to monosaccharides. Polysaccharides are multiple units of sugar, and they're broken down to single sugar units, monosaccharides. Okay, it begins in the mouth. Mouth has what's called amylase. This is an enzyme, and it starts to digest carbohydrates. So it begins in the mouth with amylase action on the starches. Continues in small intestine. Okay, after it goes in the stomach, from the stomach goes to the small intestine. The small intestine with the pancreas. Pancreas produces amylase to form monosaccharides. Okay, so it starts in the mouth, it starts to break it down into smaller sections, goes through the mouth, esophagus, stomach, to the small intestine. Once in the small intestine, pancreatic amylase starts to break it down and cleaves each unit into monosaccharides. Proteins, digestive amino acids, okay, begins in the stomach, okay, with the acid hydrolysis. Acid hydrolysis starts to chop the proteins into smaller fragments of amino acids. Then serine protease is an enzyme that acts in the small intestine and starts to cleave each into smaller amino acids, into the individual amino acids. Fats are broken down into fatty acids and glycerol. And these also begins in the small intestine with the different fat globules. Dispersed with bile salts. Bile salts come from the liver. Okay, and they help in the production or the breaking down of lipids. Okay, they degrade these lipids with pancreatic lipase. So the pancreatic also produces lipase, and it also produces amylase. Amylase breaks down polysaccharides, the sugars and polysaccharides, your carbohydrates, and it also starts to break down your fats. Your lipids into glycerol and your fatty acids. Okay. So salivary glands secrete amylase that starts to digest your starches. The stomach secretes hydrochloric acid and pepsin. Hydrochloric acid denatures a protein, starts to break them down with the acid content. And then pepsin digests these proteins. The smaller sections of it starts to break them into individual amino acids. The pancreas secretes serine proteases and lipases, which helps in the breakdown of lipids and proteins. The liver, the gallbladder deliver bile salts. Okay. The liver produces bile. The gallbladder does not. Gallbladder is just a storage unit. So a gallbladder stores bile for when it's needed, then it can release it. But the bile is actually produced in the liver. So then you have amino acids, which were your proteins, monosaccharides with your sugars, your starches, enter the cells via active transport. So the cells require ATP to actively put them into the cells. Fatty acids and glycerol, the lipid break from lipids, move via passive transport. Because remember, your cell membranes is composed of a lipid bilayer. It's a fatty membrane. So fatty acids and glycerol can move through it, whereas amino acids, which are polar, and sugars, which are polar, cannot. Okay, so fatty acids and glycerol, they come in through a passive transport. They don't require energy. But to move amino acids and monosaccharides through it, that requires energy. All right. So here's hydrolysis reactions: carbohydrates, proteins, and fats. What we were just saying. So we have got, this is a disaccharide, which means two sugars. We're going to break this bond by adding H and OH, just like we did before. Remember that 4 or 5 chapters ago? Okay, and we're going to produce monosaccharides. And the enzyme we're going to use is glycosidase. Glycosidase breaks the bond between this disaccharide, and it produces 2 monosaccharides. Now that can be absorbed. Okay. That is our carbohydrates, our sugars. Our peptides, our proteins, we break the peptide bond. Remember how we broke the bond right between the carbonyl group and the nitrogen? We added OH and H. Again, we added water, hydrolysis between those 2 bonds. And then we formed our amino acid and another amino acid. And remember, the R group is the only difference in amino acids as to what type they are. There's 20 different types. Okay. Next one is our triglyceride. Okay. Remember, this is glycerol right here. Well, with the OH is right here. Okay. This is your glycerol molecule. And then these fatty acids attached to it. This is a triglyceride. Okay. Lipase breaks down lipids, which are triglycerides, okay, into their fatty acids, okay, and glycerol. And again, you've added water, one for each fatty acid. So you've added water. Let me get rid of some of these. You've added water across this bond right here, this bond right here, and OH, hydroxylic acid, and then OH on the glycerol. So you've added H to this side to produce your carboxylic acid, and then OH to produce your glycerol. Okay. So we assimilate the smaller subunits in the pathways of energy metabolism. So the basic thing is once you eat food, whether it's a protein, carbohydrate, or fat, lipid, it's big. And you got to break it down to the small subunits to use it for energy. So breaking it down releases energy because you're breaking bonds. That releases energy. So you assimilate the small subunits into the pathways for energy metabolism. So you have major pathways of energy metabolism. So sugar enters glycolysis as glucose or fructose. Glycerol, okay, produced by the hydrolysis of fatty acids, enters glycolysis. Okay, once you break down fatty acids. Okay. Sugars are converted to acetyl coenzyme A and enter the citric acid cycle. Proteins enter many steps of it. Okay. And you have the carbon skeleton of your amino acids. And then fatty acids enter the citric acid cycle after conversion to acetyl coenzyme A, just like sugars. Okay. So these are pathways you're going to use to generate ATP. So acetyl coenzyme A carries acetyl groups, which are 2 carbon remnants of the nutrients. So you've broken down all these nutrients into these 2 carbon molecules at the end. So acetyl coenzyme A enters into the citric acid cycle. Okay, electrons and hydrogen atoms can become harvested, and the acetyl group is oxidized to produce CO2. So basically, we're taking all of our energy down, running it through the citric acid cycle to produce carbon dioxide by oxidation. Okay, so electron, hydrogen atoms and harvested are used to produce ATP during this oxidative phosphorylation. Again, we've increased the bonds to oxygen and CO2. Remember, if you look at the structure of CO2 was carbon, and we have 4 bonds now to it. Okay, so each double bond each side of the carbon molecule. So we increase the number of bonds to oxygen, it has been oxidized, and we produce a lot of ATP along the way. So briefly describe the 3 stages of catabolism. Catabolism is the breaking down of food or any food, whether it's lipids, carbohydrates, or proteins. And you need to know what all 3 of those break down into. Lipids break into glycerol and fatty acids. Proteins break down into amino acids. Carbohydrates break down into monosaccharides. Okay. Remember, catabolism gives off energy as you're breaking it down. So first stage is digestion, which is hydrolysis, adding water between the bond. Okay, occurs in the stomach and the small intestine. It starts in the mouth too. Second stage, the monosaccharides from your starches and sugars, amino acids from your proteins, and fatty acids and glycerol from your lipids are converted by metabolic reactions into molecules that can come completely oxidized. They can run through the Krebs cycle. They come into acetyl coenzyme A. Okay. So you take your monosaccharides from your sugar or starches, your amino acids from your proteins, your fatty acids, glycerol from your lipids, and you convert them by metabolic reactions into molecules that we can run through the citric acid cycle. Okay. Then the 2 carbon acetyl groups of acetyl coenzyme A is completely oxidized by the reactions inside the citric acid cycle, spins through a cycle and produces carbon dioxide. The energy of the electrons that are harvested in these oxidation reactions, and oxidation reduction reactions are moving of electrons. And when we harvest these energy of electrons as they move to different energy levels, okay, we use that to make ATP. That's how ATP is made from the food we bring into our bodies. Okay, discuss the digestion of dietary carbohydrates, lipids, and proteins. We just did. Carbohydrates break into monosaccharides. Lipids break into glycerol and fatty acids. Proteins break into amino acids as they run down. So the proteins, carbohydrates, and triglycerides in our diet are too large to be taken into the cell lining of the intestine. So you have to break them down. That's what all this is about. They must first be digested into their component subunits. So digestive enzymes hydrolyze our polysaccharides and disaccharides, those are sugars with multiple units, into monosaccharides that can be taken into the small intestine by active transport. Okay. Digestion of proteins begins in the stomach and is completed in the intestine, the small intestine. Proteins are degraded to amino acids that are also taken up by active transport. Okay. Dietary lipids, usually triglycerides, are emulsified. That's our first step by bile salts in the duodenum. Duodenum is the first part of the small intestine, right after the stomach. That's where the stomach empties its contents into. Right? So dietary lipids are triglycerides. They're emulsified. Remember, they get surrounded by these bile salts. These form these emulsifications in there. Okay? Pancreatic lipases hydrolyze the triglycerides to monoglycerides. So it starts breaking them down. And then you got glycerol and fatty acids are absorbed by intestinal cells by passive transport, simple diffusion across the membrane. So you got to remember that the glycerol and the fatty acids are nonpolar, can travel across the membrane. Okay. Ebden-Meyerhoff pathway. This is glycolysis. We'll do another 5-6 minutes and call tonight. We'll finish it, a couple more slides here. So glycolysis is a breaking down of glucose. Glycolysis, glucose breaking down. So we're breaking down glucose to give us energy, glycolysis. It's a pathway for carbohydrate catabolism. Remember, carbohydrates are sugars made up of polymers, go back into disaccharides into monosaccharides. You begin with D-glucose as your substrate. So you break everything down to D-glucose. That's what you're going to start with. All organisms can use glucose as an energy source. They can all use energy. They can all use glucose to produce ATP. Requires no oxygen. Occurs free in the cytoplasm. In other words, it doesn't cost energy for glycolysis to occur. It's a 10-step pathway catalyzed by enzymes. The products of glycolysis, you got substrate level phosphorylation. By adding phosphates to it, you get 4 ATPs generated. Okay. A phosphorus group is transferred to ADP from the 1,3-biphosphoglycerate and phosphoenol pyruvate. Okay. You transfer in an ATP is given off for that. So transfer is transferred to an ADP, and from that ADP, you make ATP. Okay. NADH carries hydride anions with 2 electrons. Pyruvate depends on cellular conditions, whether it's going to be aerobic or anaerobic. What's going to happen to pyruvate? Okay. The anaerobic oxidation. Anaerobic means no oxygen present. This occurs without the presence of oxygen. Aerobic means it includes in the presence of oxygen. Anaerobic means it includes in the absence of oxygen. So the anaerobic oxidation of glucose gives you 2 molecules of pyruvate. You have glucose plus 2 ADPs plus 2 phosphates plus 2 NAD pluses go to 2 pyruvates, 2 ATPs, 2 NADH, 2 H pluses, and 2 H2Os. Okay. Remember, it's going to 2 because pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule. So it's going from 6 carbon to 3 carbon, so you're going to get double. So it's going to take 2 ATPs and 2 PIs to give you 2 ATPs, 2 NADHs, 2 H plus, and 2 H2Os. So pyruvate is used in the following reactions to sustain glycolysis. That's what keeps glycolysis going. NADH must be reoxidized so the glycolysis can continue. So you have to reoxidize NADH to use it as NAD plus. Okay. So here's your summary of glycolysis. This is glucose. Takes 1 ATP to go to glucose 6-phosphate or fructose 6-phosphate. Okay. Then it starts down into glucose 3-phosphate. Remember, it breaks apart. That's glucose 6-phosphate to glucose 3-phosphate. Then it goes to 1,3-biophosphate glycerate. Okay, it goes from NAD plus to NADH, then ATP, ADP to ATP. So you're making ATP. And eventually it's going to break down into your pyruvate molecules. So you're producing ATP from ADP. You're storing energy as you're breaking down glucose and preparing it to form pyruvate. And there's 10 steps involved in that. Okay, you can see the 10 different steps right there. We'll go through those in a greater detail here. And that's to form pyruvate. Pyruvate is a 3-carbon molecule that can be used to form acetyl coenzyme A, which is going to enter the citric acid cycle to produce more ATP. Okay, so you prepare the glucose, making a glucose 6-phosphate or fructose 6-phosphate, and then running it through the 10 different steps you can have before it enters the Krebs cycle.